Uh, I'm Lane Smith. I'm New York Sea Grants Research Program Coordinator, and I'd like to welcome you all to our first of our virtual summer seminar series. And this is the first year that we're doing this. Today's speaker will be Dr. Chris Gobler. And we're going to be having today Dr. Chris Gobler, who will be giving a talk titled New Approaches to Decoding Brown Tide in Long Island Sound Estuaries. And we have 3 more seminars in this series. In July, we will be having Dr. Jace Bernhardt from Hofstra University, who will be giving a talk titled Developing a Virtual Reality Video Game to Simulate Rip Currents. And in August, we will be having a seminar by Dr. Scott Steinschneider at Cornell University, who will be giving a talk titled A Probabilistic Parcel Level Inundation Prediction Tool to Support Community Preparedness to Flooding Along Lake Ontario. And our final virtual seminar will be September, given by Dr. Karen Lindberg and Kayla Smith at SUNY ESF, Reconnecting Waters for Eels and River Herring, Public Perceptions of Dam Removal in the Hudson Mohawk River watershed. And we hope you can all make each of those seminars. Okay. So before we get started, I just wanted to mention that this seminar is being recorded and it will be made available on our YouTube channel. We're also streaming live simultaneoly on Facebook Live. I also want to mention just a brief overview of the seminar that we'll be hosting today. When we're on Zoom, some of you who may not be quite as familiar with using Zoom, I just wanted to just show you on my screen here. Gotta jump back and forth. So I'm sure most of you are familiar with Zoom. If you on your client, you'll see a black bar on the bottom, and if you click on chat, and then if you you'll see a chat window, and you can submit questions to the host, and I can read them out for Chris to answer. For those of you on the Facebook Live, you can submit questions on that comment section, and they'll be passed along to me to read. And in addition, you can also click on participants, and you will get a let's see if I can get this to. You will also have on participants, you'll see that you can raise your hand, and then that I can unmute you, and then you can ask your question. So if there are any questions, you can just enter them in the chat, and we'll try to answer them. Okay. Now, as we move along, I'm going to make Antoinette a co-host, and she's going to introduce our speaker, and but first give a quick uh, presentation before we start with Chris. So Antoinette, are you ready?
I am. Thank you, Lane. And good afternoon, everyone, and welcome to the first in our Grants virtual summer seminar series. Thank you so much for taking the time to join us today. Today's seminar is a continuation of an effort to facilitate harmful algal bloom outreach and education programming in the Marine District, where we know that harmful algal bloom, though it's seasonal, it is certainly problematic. Sea Grant worked jointly with Suffolk County government to organize a harmful algal bloom symposium in 2018, and that was held in Great River. It was very successful. And we actually had plans well advanced to facilitate a similar meeting, a second symposium in April of this year, but as you all might suspect, as a result of COVID, we are, I can say the rest is history. The Harmful Algal Bloom Symposium series is actually supported under an EcoHab Research Award to Dr. Chris Gobler, who we'll hear from momentarily, and EcoHab, ECOHAB, acronym is for NOAA's Ecology and Oceanography of Harmful Algal Blooms program. And that is a program that is being administered through the agency's National Centers for Coastal and Ocean Science. So for today, we have two speakers. First, we'll hear from Dr. Quay Dortch, who's the EcoHab program officer, and she will highlight a selection of programs that may be of interest to participants. And after that, we will hear from Dr. Christopher Gobler, who will spend the bulk of the remaining time talking about the new approaches to decoding brown tide in Long Island estuaries. So I'd like to thank you for your attention, and I'd also encourage you to complete the survey link that Lane will put in the chat box. Please just complete that before you leave the meeting, because we do value your opinions. And without any further ado, I will turn this over to Quay.
So can you show the first slide, my only slide?
Lane.
All right, so you want me to show it? Okay, hold on.
Anyway, while he's getting that slide up, I'll go ahead and start talking, because I don't want to take much time away from Chris. What?
See that?
Yep, I can see that.
All right.
So we in NCOS, for our HAB programs, have two major groups of programs. One is the research funding programs, which are listed here, of which Ecology and Oceanography of HABs is one of them. And these are research programs that you have to apply, submit a proposal. And one, the Ecology and Oceanography, looks at the causes and impacts of HABs. The second one, Monitoring and Event Response, is largely to build capacity within the monitoring and management community. And that's a program that I think might be of particular interest for people who might be on this seminar. And then the Prevention, Control, and Mitigation of HABs program is doing exactly what the name says, and might be a way, for example, if you wanted to test a control method and develop for your situation, you might want to think about that program. But the one that I think is most useful for everybody is the HAB Event Response Program. It's not a huge program, but within NCOS, we have both a small event response pot of money. So you can call us up one day, and depending on the situation, the funding can be arranged within hours to days to help you respond to a particular problem. But we can also put you in touch with people who can do remote sensing, also with phytoplankton identification, the Phytoplankton Monitoring Network, and also the analytical response team that can do toxin analyses. It's more oriented towards marine HABs than it is towards freshwater HABs, but there is some capacity for freshwater HABs as well. So I just wanted you to know about these programs, and those websites, if this slide gets posted, those websites will lead you to contact information for any of those programs. So I don't want to take any more time away from Chris, so that's all I really had to say, and I'm looking forward to hear what Chris has to say. Or is it going to be Chris and Sonia?
Thank you, Quay.
Thank you,
Very
Quay.
interesting. I wasn't even aware that there are so many, resources available. So I'll certainly look into that, list that you have shared with us. And I guess now, without any further ado, we'll turn this over to Dr. Gobler, who will give us an insight about the advances of understanding brown tide and how they work. Chris.
Okay. Hopefully everybody can hear me.
We can hear you, Chris.
Great. Thank you, Lane. Okay. Well, Quay made a good point. Yes, it's going to be just Chris or Chris and Sonia, and so I think I'm giving the presentation, but I do want to, from the get-go right here, acknowledge a great collaborator of mine who's on the Zoom call, and when we get tough questions in the end, I'll ask Sonia to answer them. But Sonia and I have been collaborating for quite some time. I remember fondly in 2006 when we first met. And she said, oh, well, I'm starting to do brown tide research because I heard the brown tide genome is being sequenced. And that's an effort that, for a proposal I put into the Department of Energy to sequence the brown tide genome. And that really began what is close to a 15-year collaboration. And it's exciting that we've been able to continue that collaboration and make some great discoveries along the way. And I'll seek to share those with you today. And this both speaks to a current EcoHab project that we have or that we're wrapping up, as well as a little bit of a history of the work we've done on the brown tide and via genomics and transcriptomics. But just as a way of introduction, we've heard the term harmful algal bloom. So this, of course, is an official term to define when you have the growth of an algal species with density that can have a negative impact on human health and or an ecosystem. And so brown tide is just one of many. And of course, these are both marine and freshwater, has been alluded to already. And the most common type of HAB that people are worried about are the ones that make biotoxins. Because those toxins can accumulate in shellfish, people can eat those shellfish and get sick. There's certainly other exposure routes as well. Brown tide is not one of these types of harmful algal blooms. Instead, it fits in the category of what we call ecosystem disruptive harmful algal blooms. That can cause the death of marine life. Here I'm showing a fish kill, which can be due to HABs, probably not due to brown tides per se. But these can be quite costly events as well. With regards to brown tides, you can see from this picture how they get their name. They are dense accumulations of small cells, literally billions of cells per liter when they get quite dense. And you can see the micrograph of them there, fairly non-distinct. You can see some more micrographs here, an SEM and a TEM on the left and right respectively. These are small, what are known as pelagophytes, which are fairly rare types of phytoplankton. Interestingly, pelagophytes are ubiquitous in the global ocean. But there's just two species that cause harmful brown tides, and the one I'm going to talk about today is known as Oreococcus anaphygeferens. And I'll just show you that these had never been identified by scientists before 1985. And then suddenly, in the summer of 1985, waters across New York, Rhode Island, and New Jersey all suddenly turned very, very brown, and there started being collapses of bay scallops, and those were the first brown tide blooms caused by Oreococcus anaphygeferens. Since that time, there have been other brown tides identified by other species, the related species Oreo umbra, other brown tides in other locations, a southern expansion of Oriococcus in the U.S., as well as an expansion to South Africa. Last decade, it was first identified in China, and in fact, the blooms in China are actually the biggest and most economically devastating ever recorded compared to the ones in the U.S. And then the other brown tide species, Aurea umbra, has been spreading this decade. Focusing in on the U.S. East Coast, this figure here shows you the locations where the brown tide cells have been detected, and then also where blooms have been recorded. And really, Long Island and New York has been the epicenter for these events. They've occurred most consistently on Long Island, the first bloom in 1985. And, since then, there's maybe been, a handful of years without a brown tide. It's really recurred every single year. In the first years, it was more on the eastern end of Long Island, on the South Shore, and they've stopped occurring there since 1995, but they've been a chronic issue across the South Shore of Long Island since then. And again, this is an ecosystem disruptive harmful algal bloom, and the brown tide has been directly and indirectly related to some of the most severe collapses of fisheries in the history of Long Island. So, for example, the bay scallop fishery on eastern Long Island was the largest scallop fishery on the east coast of the U.S., and its collapse in 1985 was definitely due to the reoccurrence, in fact, 3 consecutive years of brown tides. As an organism that only lives 2 years, this is definitely this was devastating to that particular fishery. You can see a small recovery in 1994, and then in 95, another very severe bloom that led to the collapse. There's been recovery since then as brown tides stopped occurring in this estuary. We know the hard clam was historically one of the most important fisheries in the history of New York State. This original decline or initial decline through the 80s, early 80s at least, was not due to brown tide. It's due to overharvesting that's been established scientifically. But the inability of this fishery to recover, despite decades of attempts to revive the fishery, has definitely been due to the recurrence of brown tide year after year. And we also know that seagrass beds across Long Island have retreated significantly due to brown tide, and that the aerial extent of seagrass meadows is much less than it was before we started having brown tides. And it's unfortunate to report that actually, as of this week, the brown tide is back on Long Island. We've been monitoring the brown tide in the South Shore of Long Island. I actually thought we might be out of the woods with regards to brown tide in 2020 because levels declined a little bit in early June, but they popped way up this week to over 300,000 cells per liter in Great South Bay in the vicinity of Patchogue. And we know that only 35,000 cells per milliliter, is when they start having a negative effect. And I'll just briefly divert and mention Sea Grant. If you see water like this, Sea Grant has just initiated an ability for citizens to report on harmful algal blooms, specifically marine harmful algal blooms. And so you can go to their website and you can log in there, and within that site you can then go in and fill out a form and indicate where your observation is being made and what you're seeing, and that will be followed up on by Suffolk County, and perhaps my laboratory will be involved as well. And that this information will be used to guide future decisions and will allow for continued mapping and understanding of the where and when of HABs. Mike Jensen is helping head that up. He's part of Suffolk County Department of Health Services. But back to the brown tide, and really the topic at hand is, and what the rest of the seminar will be about, is why do we have these brown tide blooms? You know, why is it that one week the water in Great South Bay might have looked like this with many different phytoplankton, and the next week suddenly we have the system taken over by brown tide. And the approach that Sonia and I have used over the last 2 decades to try to understand this is really what we call genome enabled. And so I mentioned that when I first met Sonia, we had our proposal approved to sequence the Oreococcus genome. That actually happened late last decade. In 2011, we published the paper on the first harmful algal bloom genome, specifically on Oreococcus. And that was an exciting outcome. I won't go through the details, but just highlight some of the things we found with regards to the Oreococcus genome. Surprisingly, despite the fact that it's a very small algae, just 2 microns, its genome was actually larger than the other types of phytoplankton it was competing against, and it contained more genes than the other algae. Which led to the hypothesis maybe these genes, it has a bigger toolbox in order to deal with environmental conditions. When we looked at the genes that it was enriched in, that the genes that had more of than the competing species, it had many more genes associated with adapting to low light levels and to growing and using organic matter. And then interestingly, we also found a series of genes that were not present at all in the competing species that we hypothesized may be involved in predator defense, specifically against things like zooplankton, which are the primary predators of harmful. So having found these genes in the genome, the next step for us was to understand, are these genes important during when the brown tide is actually growing? And that's both in culture, when we grow it in the lab, but also during the blooms. Because of course a genome is just sort of what the potential for the organism is to do. It's not necessarily that they're using those genes or turning those genes on and making the proteins associated with them during bloom. Some genes may be inactive or they may be always active. The question is, do specific genes turn on specifically to facilitate the occurrence of a bloom? And so we specifically, one of the first things we did was to conduct some laboratory experiments where we would take a culture and then grew it under conditions that we knew would be commonly encountered in an ecosystem setting. So that is growing it with low light or low nitrogen or low phosphorus. And then with the help of one of Sonia's PhD students, we identified different genes that turned on in response to different environmental conditions. So genes that are specifically responding to low nitrogen conditions, and genes that are specifically responding to low phosphorus conditions, and those that are specifically responding to low light. So this was a great tool to have because we, having done this in a controlled laboratory experiment, we now had the opportunity to go actually into an ecosystem setting and determine if indeed the things that we can turn on and off in a lab setting could also be turned on and off, or are they turned on and off during a brown tide within an ecosystem setting. So are these gene expression patterns continuing during or observed during brown tides? And so the most common location we worked in to answer these questions is Quantuck Bay. It's a small embayment on the south shore of Long Island, located equidistant between Shinnecock Bay and Moriches Bay. And it's been a great bloom laboratory. It's predictably had blooms. And when blooms occur, the blooms in Quantuck Bay often seed and spread throughout the neighboring bays. So not only is it continually having bays, but it seems to be the source of other estuaries in the region as well. So one of the first follow-up experiments we did was looking at some of these phosphorus responsive genes. And one of the most well-made, if you will, genes for this is a phosphorus transporter. So transporters, many of these genes, in fact, are transporters. They're at the interface between the cell and the ecosystem. And therefore, they're the most responsive within the organism and the most likely genes to turn on or off in response to an ecosystem condition. So I'm showing you a bloom here in Quantuck Bay that you can see took off and grew very quickly in early June. peaked in mid to late June and then began to decline. I'm showing you in parallel the dynamics of orthophosphate. And this pattern that you see for phosphate is quite common. Levels are usually low early in the season, and then as sediments become anoxic, which is very common in coastal environments, they become low oxygen temperatures, ramp up, the phosphorus is released out of the sediments and the levels are high. And what's interesting is when we look at this pattern and we compare it to this particular phosphate transporter, what we found during one of these earlier blooms is very high expression levels of this phosphate transporter in the beginning stages of the bloom. And then as the phosphorus levels get higher, the bloom seem to, or the brown tide seems to no longer need to produce a lot of these proteins, and therefore gene expression levels go down. And so you could almost see that once you hit a certain concentration of phosphorus, the gene is silenced and no longer being expressed. And so potentially, again, indicating that the gene is being turned on and off as a response to ecosystem conditions. In a follow-up study, again in Quantuck Bay, we again examined the occurrence of a brown tide that had, again, very similar dynamics. So a peak ramping up in June and declining by the end of June. And then in this particular case, we're showing the ratios of the inorganic nitrogen to phosphorus. And Alfred Redfield determined, and Woods Holder from the graphic in the middle of the 20th century, that this ratio should be about 16. So the ratio as it is shown here would say that there's potentially not enough phosphorus. Just looking at, we collected transcriptome samples from the bloom on these 3 dates here. When we look at those 3 dates, and then we look at all the different classifications of genes here, the different gene pathways, we see, well, this is the pattern we see. And so firstly, what this is showing, these different pathways are called KEGG pathways. They're related genes and different biochemical pathways within the Oreococcus. And as a heat map, it's showing you the where we're seeing most of the reads in the red and fewer reads in the blue. And so a few things to take away here. Firstly, you can see there are distinct differences on each date. So genes are being turned on and off as the bloom progresses. And then when we look at the different samples being taken, these were taken not too far apart in the bloom, sort of at the peak and maybe the beginning of the decline. Many of these strongly expressed pathways are what we'd call heterotrophic pathways. So things associated, for example, with carbohydrate metabolism, proteases for degrading proteins, protein processing, and amino acid metabolism. Beyond just looking at these 3 dates in this study, we also looked at a particular experiment. And this particular experiment, conducted here in June, showed that when we added nitrogen, we didn't get a strong effect on its own. When we added phosphorus, however, there was a growth effect. And as you can see here, there was actually an additive effect or even synergistic effect for nitrogen and phosphorus. That is, the outcome with regards to phosphorus and nitrogen together was greater than we would have predicted from phosphorus alone. So this suggests to us that actually both compounds are important for the bloom. But just focusing on the nitrogen and phosphorus, this is a little bit of a complicated plot, but essentially it's showing all the transcripts of the expressed genes from Oreococcus, and it's comparing on the x-axis the control treatment, on the y-axis the nitrogen addition treatment. And on the one-to-one line means the genes are not different between the control and the nitrogen. And so essentially that's what this figure is saying. When we added nitrogen, we had a single gene here that was upregulated, and that is specifically the, you can see the ribonucleotide reductase gene. But when we compare the phosphorus treatment, shown here, to the control treatment, you can see there are multiple genes expressed here that are at lower abundance, they're below the line. And what that means is when we added phosphorus, all of these genes associated with phosphorus acquisition were turned off. right? So that, and stated another way, this experiment is telling us that these genes are turned on during the bloom to help Oreococcus get more phosphorus. By adding that phosphorus back, you can turn them off. We do see here, however, a urea transporter that's upregulated. And so what that means is that urea is a type of nitrogen. And when there is more phosphorus, it's working harder to get nitrogen, and specifically in this case, urea. But again, the takeaway message here, again, particularly when you see the growth response and the synergistic effect of nitrogen and phosphorus together, supports a conclusion out of the EPA that was made a few years ago that to prevent eutrophication, coastal ecosystem and coastal management should focus on a dual nutrient criteria, thinking about both nitrogen and phosphorus. The final points I'll make today are with regards to the data set we're just finishing up on or trying to wrap up on from a bloom several years ago, again in Quantuck Bay. It looks a lot like the other blooms, but there are some differences in that this bloom was definitely longer lasting. So you'll recall the other blooms sort of peaked in June and then they were done by July. This one, as you can see, was an extended bloom, more than 1 month at over 1 million cells per milliliter, 1 billion cells per liter. This is one of those really damaging brown tides. So it grew up quickly and it was sustained for a long period of time. And we were very fortunate that we began sampling when it almost wasn't even present, and we're able to sample all the way through. And for this particular project, we were not just interested in Oreococcus, but we also wanted to understand from a transcriptomic point of view how other competing phytoplanktons, in this case Synechococcus, are behaving during the bloom and how their transcriptomes may also be changing. But to look at this bloom, we can break it up into 3 parts: the initiation phase, the peak phase, and then the decline. And what I'll point out to you here is that for each of these phases, we calculated a growth rate. And so what you see is there's fairly rapid growth during the first half of June, and then sort of this sustained level of not a very high growth rate of 0.05 per day, and then obviously the decline. I'll talk more about it later, but Sonia pointed out to me today, and it's important that I emphasize now, is that even though you don't see a lot of growth here, what is important is that what we see is actually the balance between the growth of the cells and the mortality of the cells. I'll emphasize this later, but just because they're growing at maybe a slower rate, that doesn't mean that the cells aren't growing. It just simply means that they're the balance between growth and grazing are perhaps is tighter than it was perhaps earlier on in this early stage. But again, we were very fortunate in that every single week we were out there, we were able to collect samples to sequence not just the transcriptome of Oreococcus, but also of Synechococcus, and we will also have the opportunity to look at other phytoplankton out there. And in fact, so we've done that. And when we look at the transcriptomes, you can see here, there's a whole host of other phytoplankton that we're getting good sequence reads to. So this is each individual sample we collected over time. And what it's showing is what percentage of the transcriptomic reads that we're getting map to Oreococcus versus other well-known, in this case, we'll call them eukaryotic phytoplankton. So the cyanobacteria wouldn't show up, the Synechococcus wouldn't show up. But you can see in those dates, as the bloom is ramping up and at its peak, it's representing more than 80% of the total transcriptomic reads. And then you can see this radical shift later on, and then maybe a little bit of a rebound later in the summer. When we look at the transcriptome of Oreococcus over time, this is similar to the plot we've already shown. So firstly, in very good news, we see that we're picking up about 85% of the genes in the Oreococcus genome via our sequencing effort. And that's really, really good because we know that both our samples are good and our informatic, we're informatically covering getting response out of the environment as well. We also see that these transcriptomes are very dynamic. So you can see here, I mean, let me just say that along the y-axis here, each of the different lines are different pathways and or genes. And I haven't said yet, but let me say this now. There are more than 11,500 genes in the Oriococcus genome. So this gets to be a very complex process. And it's always a struggle in these sorts of studies on how deep to go and how broad to be. And so clearly, I could, if I were to talk about all these genes, we'd be here for weeks and weeks. So in this case, we're taking a very, very broad view. So you can see we're clustering the most similar genes here. And but what I'd like to show you is how, if you look at these different phases of the bloom, again, these different growth rates that we were able to calculate during the bloom, you can see that that initiation phase, just these first two samples, look very, very different than the next phases of the bloom, particularly the sustained difference. And so you can see all these genes that were in low abundance are suddenly in high abundance, and conversely, genes that are in very high abundance during the peak of the bloom are in low abundance. And so again, just to take a broad view, what we seem to be seeing is a different groups of genes responding differently and being expressed differently at different phases of the bloom. So during this initiation phase, the things that are turned on in high levels are genes associated with nucleotide synthesis, DNA replication, the cell cycle is quite active, amino acids are being synthesized. And so it's pretty exciting that actually we can dive into the genome here, not just genome, but the transcriptome, what genes are being expressed. And we're seeing that they're the ones associated with rapid growth. And then conversely, when we get to this peak stage, these things like DNA replication and cell cycle are turned off. And instead, we're seeing things like fatty acid synthesis and then lipid storage. So it's sort of a maintenance stage of the cell, and the cell deciding that it's going to take the resources it can get and put them into storage so it can exist in this maintenance stage. And again, this is a bloom, remember, that persisted at over 1 million cells per milliliter for more than a month. And it did that by slowing down the DNA replication and growth, and instead going to this maintenance via lipid synthesis and storage. But there was continual photosynthesis and ribosomal genes continuing to be expressed as well. So now we can go in and ask the nutrient story. Again, this is a different bloom from before, but in this early experiment that I'm showing here, one that was conducted in early June, here I'm showing the responses from many different phytoplankton groups, but if we just focus on Oriococcus, in this early stage of the bloom, again, phosphorus seems to be important. So that's consistent with our prior work. But interestingly, during the peak of the bloom, and we saw this many times, we actually saw a nitrogen response. And so again, this may be related to there being more phosphorus in the ecosystem as the bloom progresses. But nitrogen ended up being more important during the peak of the bloom and during the sustenance period. So this is just one experiment at the peak, but other experiments on either side of the shoulder here also showed a nitrogen response. Now, I showed this figure earlier whereby we were able to identify specific genes that are turned on and off with regards to nitrogen limitation and phosphorus limitation. And so, Sonia went in and looked at those particular genes and was able to prepare this figure here that, again, it breaks up the bloom into the initiation stages, the peak, and then the decline. And here we have this is looking at genes associated with low phosphorus and low nitrogen. So the genes, again, here, that they would turn on to deal with either phosphorus stress or nitrogen stress. And what we see, actually consistent with our experiments, is during that initiation phase, we see that there's an upregulation of genes associated with phosphorus acquisition. So, and if we look at the total amount of phosphorus here, you can see that's the lowest levels early on in the bloom. So those phosphorus acquisition genes are turned on. But when we transition to the peak of the bloom, those genes are silenced. The 1-to-1 line means there's really no difference between the expression of those genes in that treatment versus the control. However, again, consistent with our experiments, when we look at the peak of the bloom, we actually see these nitrogen acquisition genes, or genes associated with nitrogen limitation, being turned on. And so the bloom, therefore, transitioning from phosphorus limitation to nitrogen limitation. And this is a very clear result in that we were able to demonstrate this not just from experiments and changes in cell densities, but in looking at the genes that over the years we've shown are indicative of phosphorus stress or nitrogen stress. Those genes are telling us that as well. So literally these cells are telling us we don't have enough phosphorus early on in the bloom. And later on in the bloom, they're telling us we don't have enough nitrogen. So again, it hurts back up the same idea that you really do need to think about both nutrients. The very last thing I want to talk about is predation, and specifically zooplankton grazing. This is a slide I actually use in my introductory phytoplankton ecology class at Stony Brook University. It's simply just saying that the number of cells you see in a bloom are going to be a function of the balance between the growth of the cells and the mortality of the cells. And through the years, it's been demonstrated that one of the reasons brown tides do well is that they're not grazed at as high a rate as other phytoplankton are during brown tides by zooplankton, their main predators. And so I actually showed this figure before, and I purposely sort of skimmed over some of the details here. But in these experiments where we had a control, we had a nitrogen or phosphorus or both, we also had a treatment, it's a little obscured here, where we added a grazer, specifically Oxyrius marina, an organism that we know can consume the brown tide. Now, what's actually interesting is that if you look at the growth response, we added more of this grazer, and yet the growth rates went up. So here's our control where we added nothing. Adding the grazer actually elevated the growth rate of the species. And this is in direct contrast to some of these other cyanobacteria here, where you can see if you compare the control of the grazer here, they're actually being consumed. So something's going on here where this grazer is introduced into this ecosystem setting, and it's making an active decision to not graze on Oreococcus and rather go after other organisms. Now that's interesting in and of itself, but we also then looked at the gene expression patterns. So these are plots sometimes called manta plots. They're kind of complicated, so I'm just going to make it super simple. And you can see there's a 0 line here, and anything right on that line means that when you compare the individual treatments I'm showing here to the control, if it's on the line, they're no different between those two. And to make it even more simple, they're color-coded. So anything that's red means that the genes are either being up or down regulated significantly. So statistically significant change in expression patterns, or I should say transcript abundance. And so the takeaway, as you look at these dot plots, what you really just want to look at is say, where do I see a lot of red dots? And the answer very clearly is the zooplankton addition. So when we added the zooplankton here, you can see the Oreococcus transcriptome got very, very active with literally thousands of genes being turned on and turned off. Statistically, if we did something called the principal component analysis and looked at each of the replications, of course, these are carefully replicated experiments with triplicates for each. When we do this sort of comparison, principal component analysis, we see the grazer experimental bottles, the transcriptomes in those bottles are very different than all the other treatments. And again, when we look at the individual gene pathways, we see that the response of the genes to the grazers, things being turned down or turned off, lower abundance or higher abundance for these transcripts, what you see out of the grazers is quite different than the response that you get from nutrients. And then finally, when we look at some of the genes that we originally identified in the genome paper back in 2011, and we hone in on some of the genes that were unique to Oreococcus, we see genes that we identified that could be associated with the brown tide being toxic or noxious to other species, as well as genes associated with light harvesting and perhaps with metals being at higher abundances relative to control, but then other gene sets being at lower abundances. So clearly, this grazer addition had a big effect, not just on the abundance of the cells, but also on transcriptomes. With that, I will wrap up. So concluding, the metabolism of and the factors controlling brown tides clearly change over the course of the bloom. And I was able to show you that during the initiation stages, we're seeing very rapid growth of Boreococcus. It's definitely outpacing the grazing rates. And then within the transcriptome, we're seeing gene sets associated with DNA synthesis, amino acid synthesis, and a phosphorus limitation signal. But when we get to the maintenance stage of the bloom, and again, remember the bloom we showed you lasting 1 month, and that's where the ecosystem damage really can set in. The growth is being balanced by grazing. We're seeing Boreococcus synthesizing and storing lipids and a strong nitrogen limitation signal. And so, clearly from a management end, and that's one of the goals of this project, is to try to understand how to best and better manage brown tides, we do need to think about both nitrogen and phosphorus. And when we think about brown tides, we also need to appreciate that it's more than simply what we call biogeochemistry or the nutrients, because there's also the balance of these grazings and grazing. And so clearly that one grazer that we added, the Alcerius marina, elicited a very strong transcriptomic response in brown tide. And it may well be that it's these grazer defense genes that are being turned on that allow the brown tide to persist for a long period. So with that, I am now done. So I would just like to thank members of the Gobler Lab for their support and getting the samples and continued support through the analysis stage. And I'll also mention the Dyerman Lab. There's been many people like Matt Harkey and Sheehan Haley and Louis Wersch. Showed many of Louis' papers, and Kyle Frischkorn, who also showed one of his papers. And also clearly the support from New York Sea Grant through the years. My very first grant that I ever got as a faculty member in the first months of being a faculty member was from New York Sea Grant to study specifically brown tide. And the very strong support from NOAA's EcoHab program. Clay Dortch gave a great overview to that, and they've been a key supporter of my brown tide and harmful algal bloom research through the years as well. So with that, I'm happy to move on to the questions phase.
Thank you, Chris. That was an excellent presentation. Thank you to you and your lab. And before we get into the questions, I just want to remind people that we have a survey for you to fill out when we're all done. If you look at your chat window, I am posting, I'm going to post the link to that chat, to that survey. Look in your chat, there you'll see a link, you can click on it, and it'll take you to a form. And it would help us out if you could fill out that survey so we can improve the seminar series for the future, and it'll help us with our future planning. So for those of you on Facebook Live, you could post a comment, and then Paul will will message me and we'll read that question. And for those of you here, you can use your participants window and raise your hand, or you could type your question into the chat box. And with that, we'll start with any questions. If you wish to unmute yourself, you can do that as well.
This is where I'll unmute and ask Chris the first question.
All right, Greg.
So Chris, I'm very interested in the timing of your phosphorus addition response and your nitrogen addition response. And I'm curious if you have correlated that with your KEGG maps to see if what's happening is that initially the cells are energy limited and are focusing on phosphorus acquisition. And then as they get enough energy, they start using that phosphorus for amino acid biosynthesis and focus on nitrogen acquisition.
Well, I mean, your narrative there is a good one. Is your question how long the experiments go, or
Well, do you have, for example, KEGMAP data or something like that that you could correlate at those different times, or were your times just too different?
Yeah, no, I mean, so the way we have the keg maps from during the bloom. We collect the samples, and literally they were within liquid nitrogen, and therefore frozen. Their transcriptomes frozen in place within minutes of being pulled out of the bay. Our experiments went for 24 hours, and then similarly, when we were done and we were ready to quantify the cells, we also froze the transcriptomes in place and sequenced those. So we do know, how they were responding to the nutrients as well. And interestingly, by and large, we saw the different growth responses, but the gene expression patterns, there were some exceptions, but they really fit over time what was happening within the ecosystem. So it seems that once they got into that extended maintenance phase, even with adding, certainly some genes are being turned on and off with regards to nitrogen and phosphorus acquisition, but these overarching pathways, they seem to be in that maintenance phase, and that seemed to continue despite adding the nutrients and such. So I'm not sure that fully answers your question, but at least it gives you a little further insight. And of course, Greg, you're happy to pick up the phone and we can talk about this further anytime.
All right, we have a question from Quay. Do other zooplankton elicit a similar response? I am not sure what other zoops graze on brown tide, but would mesodinium or copepod cause the same response?
Wow, Quay, you should be a member of my lab, I think. I have a master's degree student named Walter DeWittiak, and he's going to follow up on this particular experiment specifically by looking exactly at those two grazers. He wants to look at mesodinium, and he wants to look at, with regards to copepods, the adult phases are too large to feed on brown tide, but work that I had done with Darcy Lonsdale years ago has shown that the nauplii, the first stages of, say, for example, a Carchatonza, are able to graze on Oriococcus. So we are going to try to better decode what was going on there and what is going on there. We're going to be following up with some additional lab and field experiments.
Excellent. Don't be shy. Antoinette.
Thank
Go ahead,
you.
Antoinette.
Yes, thanks. So, Chris, I have a question. As a manager, not that I'm one, what phase of these blooms are most vulnerable and likely to try to tackle from a management perspective? Because it seems a very complex process. And at one point you start off, then you have that sustained part, and then it's like there's synergism that, perpetuates the problem. In your opinion, from a management perspective, what would be, a vulnerable phase that we could maybe target more research? Thanks.
I mean, again, clearly it seems to shift over the course of the bloom. The phosphorus inputs are coming out of the sediment, and that's really complicated. Getting the phosphorus out of for example, the entire south shore of Long Island would be quite a project. and there are, as you also probably are aware, there are efforts underway now to mitigate nitrogen. And again, following what we found is it looks like that could be effective in cap intensity of the bloom in the long term. So I think if there are opportunities to limit phosphorus inputs, that can be done, and the ongoing nitrogen mitigation approaches will help assure that in the future they don't necessarily reach the same maximum cell densities, which are really what can be so damaging and devastating.
All right, we have a question from Monica. Go ahead.
Hi, can you hear me?
Yep.
Yes.
Hi, Chris.
Hey, Monica.
Enjoyed your seminar. I was wondering if you've done any experiments to look at Orococcus gene expression responses to viral attack.
That's a great question, and I'd be remiss if I didn't mention to the audience that Monica Purcell was there for the very first brown tides and did the most important research in identifying the effects of brown tides on scallops and other bivalves. So it's actually a great pleasure and honor to have her watching my seminar. And on the virus front, great question, Monica. Sonia and I have an active collaboration with Steve Wilhelm. He's at the University of Tennessee, and we've already published some papers on on tracking the dynamics of the viral gene expression during brown tides, and in fact, just in the last week, we're working on getting him to look further into that for the bloom that I just showed you, because we want to understand not just the zooplankton effects, but you're right on the money that viruses are probably another or the other major loss mechanism during brown tides. And so towards that end, we're hoping that Steve Wilhelm and his lab group can bring more insight into the role of viruses and virulysis over the course of this bloom as well.
Great, thanks. Thanks a lot.
Thanks for attending.
All right, Chris, we have a question from David. Knowing what is creating all this, how do we begin to control the feedstock going into the bay?
Okay, well, I would be guessing at the, not fully certain what the term feedstock is referring to here. So
is this related maybe to the oyster shellfish or bay scallops maybe? David, if you can unmute yourself and maybe expand on that. I'm trying to find his. Looking for him on here. So David, I unmuted you. So if you want to expand on your question, that would be great.
Yeah, Chris, this is Dave. Just in regards to the phosphorus and the nitrogen, that is, in fact, we seem to be all agreeing that that is the major cause of this algae bloom brown tide. So, with your team and all the other people that are putting these efforts together from the science standpoint, I'm just wondering what you guys are looking at in regards to whether it be the county or others that, I know we're trying to work on some septic programs and all these other things, but ultimately controlling that coming into these estuaries clearly seems to be the most important thing. Unless, of course, you guys are working on the secret magic pill that you drop out of a plane and it's going to, eliminate all of this. Just like a penicillin pill. But, what's your team's view on that? I mean, because it seems as though that we're kind of all up against a pretty big battle here.
Sure. Well, there is no as I think you're saying in jest, there is no magic pill here. And as we move through things, as is the case for many of these algal blooms, there's only limited managerial approaches that are effective. And yes, so it's going to be mitigating the nutrients. And again, Suffolk County has begun its Reclaim Our Waters program, which is an ambitious program to upgrade and convert the septic systems for hundreds of thousands of residents from traditional systems that leach more than or up to 100 milligrams per liter of nitrogen to systems that release much lower levels. And so I think those sorts of things are going to be the key in the long haul. And I think there also is room for in the water mitigation approaches as well. And I know there are efforts, for example, at the state level, there's the New York State Shellfish Restoration Program, which is partly aimed towards enhancing ecosystem filtration capacity. It's sort of a two-sided coin with brown tides. They inhibit the feeding of bivalves, but only once they get to a high density. And at lower densities, when there's lots of bivalve feeding, they can actually prevent brown tide. So, and there's also efforts underway now, for example, for seaweed aquaculture to take nutrients out of the water as the organisms are growing. So, those sorts of in the water mitigation approaches may be a complement to watershed-based approaches that focus on nitrogen and phosphorus.
All right, so we have another question from Quay. Following up on the previous question, do clams or scallops stimulate blooms in the same way so that trying to restock them is even more futile than would be expected?
Well, again, I kind of hit that on the last point, and that is that there's a balance. And so when the bloom at cell densities below about 50,000 cells per milliliter, the brown tide can actually, the clams and oysters can actually filter out brown tide. And oysters may be even a little bit less sensitive. So, that may be an effective management approach, choosing locations correctly. And so the Long Island Shellfish Restoration Program, it specifically targeted regions that are not the epicenter of blooms, but rather sort of on the margins where if they were exposed to brown tide, it wouldn't be the same intensity as a place like Quantuck Bay. So I think that's an additional tool in the toolbox that may be effective.
All right. Somebody wanted to know if the master student, Walter Dawidiak, is the Walter Dawidiak that is known in the community, or is it a new person?
Actually, it's his son.
It's his son. All right, way to go, following the family.
Yeah, I didn't realize. I forgot I was tipping that off, but yeah.
Excellent. And all right, Maureen has a question. Have you looked at transcriptome activity when lysed Oreococcus cells are added, a false bloom demise?
No, we did, however, look at the effects of adding other lysate from other phytoplankton as a source of organic matter under the hypothesis that the brown tide would elicit a strong effect from the brown tide. And at least at the concentrations of the algal lysate that we used, we didn't see a very strong response. But the caveat being that the concentrations were moderate, and there's other ways to look at that. But we did not look at Oreococcus lysate.
All right, we have a question from T. Freeman. What is the risk to people that swim in these tides?
So thankfully, minimal. For brown tides, in particular, they do not synthesize a toxin. unlike some of the other harmful algal blooms we have in Long Island that cause things like paralytic shellfish poisoning or diuretic shellfish poisoning, blooms of brown tide or Rhecoccus are not a human health threat.
Excellent. All right, we have a question from Facebook Live. Jesse says, thank you to all the panelists. This is great. Wondering about the new ocean inlet, if it's still in effect and having a flushing impact. Also, does flushing ultimately help, or are there other helpful ways of flushing that occur through the season?
Okay, well, that's a great question. And the answers are yes, the new inlet in Great South Bay that's due south of Bellport, a place that was called Old Inlet, actually does still exist. It was formed on October 29th, 2012 by Hurricane Sandy. It's gone through expansions and contractions, but it's still there. When it occurred, I had hypothesized that that would end brown tides in Great South Bay. But interestingly, that wasn't the case. And in fact, if you look at the data, you could argue that it's made brown tides more common, but not by the inlet. And so the region just by the inlet in Bellport Bay, the water quality has certainly improved. There's better flushing, there's lower nitrogen, there's lower chlorophyll, there's more light, there's some recovery of shellfish. But areas to the west, the middle of the bay, are not really affected and actually have seemingly become more prone to brown tides. And so, you know, that's an important outcome, therefore, that while, yes, flushing helps with regards to brown tides, changes in estuarine flushing on the South Shore of Long Island cannot be predicted a priori. And so, again, the hypothesis was more flushing will improve water quality uniformly. And instead, the outcome was more flushing improved water quality in the direct vicinity of that new inlet, but actually led to poorer water quality in adjacent water. So there is the law of unintended consequences when it comes to altering flushing. And again, we can't necessarily predict the outcomes in advance.
Okay, hold on. Roger Tolson has a question. Go ahead, Roger.
Yes, hi, Chris. It's Roger Tolson. Can you hear me?
Yes.
Yeah, sure. There's been a long association with brown tide and droughts. And how does drought ostensibly prevents rain from getting into putting hydraulic pressure onto the land, and ostensibly would mean, I believe, less nitrogen getting in the bay, and we're in a drought now. Based on that observation, that droughts are related to brown tide, how does that relate to your gene evaluations?
So I think, you know, the one thing to emphasize there is that the drought brown tide relationship was strongest for the Peconic estuary during the first 10 years of brown tides there, so 85 to 95. And Julie LaRoche and others who were at the time at Brookhaven National Labs published a paper that definitely showed that, as you just stated, Roger, under drought conditions, it seems like the brown tide was more likely to occur, and that was related to the balance between organic and inorganic nitrogen, with the drought years leading to higher amounts of organic nitrogen but lower amounts of inorganic nitrogen. But since 1995, there really has not been any brown tides on the east end. And in fact, I remember well that in the late 90s, there was a prediction that, well, there's a terrible drought and we should expect a brown tide in the Peconic estuary, but it didn't occur. And also since 1995, and that's a long time, right? We're talking about 25 years, the brown tides have been only on the South Shore, and they've occurred just about every year. There's about, I think, maybe 4 years it hasn't occurred. But the bottom line, when you have that frequency, unlike what was happening in the Peconics, there's been brown tides during droughts. There's been brown tides during heavy rainfall years. So that relationship doesn't work out and has not applied to where the brown tides have been occurring for the last 2.5 decades being the South Shore. Yeah, no doubt that it probably has an organizing effect in the dynamics of the blooms, but I think it's significantly weaker than it was in the Peconics. And that may well be because the shallow nature of the South Shore means that there's a stronger coupling from organic nitrogen in the sediments. And so a more ready source of nitrogen that's being supplied.
Okay, Monica, did you have another question?
Yes, can you hear me?
Yeah.
Yes.
Yes. Yes, I was curious if these days you have currently a toxic and non-toxic strain of Orococcus, so that you could look at the gene expression differences between these.
So yes, although in my mind it's somewhat weakly defined. Monica, you of course, again, I have to give kudos to you because you were the first person to identify that indeed in culture there are strains that are more and less toxic, and seemingly that some of the first strains isolated in 1985 have become fairly benign compared to ones that were isolated, say, 10 years later. The most recent investigation to this was by one of my students, Matthew Harkey, who again looked at the same strains that you had looked at, Monica, and was able to affirm that there are some strains that are more toxic to, in his case, he was looking at hard clams, and more toxic to those than others. We've been focusing most recently on the on what we believe to be the most toxic of the strains, and I believe isolated from Great South Bay in 1995. But there really hasn't been a lot of recent cultures, and therefore, which we'd assume would be even more noxious if the reason for others being non-toxic because they're in culture for a long time. So we are focused on those more recently and presumably more toxic strains when we do our experiments, at least with things like zooplankton.
Yeah, I guess what the 95 strains have also lost their toxicity, we found. So the trick would be to get a fresh isolate from this 220 bloom. Do you have any plans to do that?
We could try. Certainly, that would be a good idea. I guess we'll wait and see how intense the bloom gets. Isolation usually works best when you've got very high density. So we'll see where the bloom goes.
Right. Thanks.
Okay, we have a question from Holly. During the summer, what is the best media source for obtaining public beach water quality?
That's a good question. I think little off topic here, but I guess I'll say two things. One, the Suffolk County and Nassau County health departments have alert systems and websites that if a beach is ever not usable because of contamination, they state that on their websites. And I guess it's a little early to say, but I will say that my lab group and other people at the university are working on a phone app that will make those sorts of things and other alert systems even easier for the public. So stay tuned for that.
Okay, I think also would News 12 be a good source?
Well, I think News 12. If you're interested in beaches, I think again the county health departments are responsible for ensuring that beaches, the water of the beaches is adequate for swimming. So I think that's probably the best. For bathing beaches, I would say that's the best source. But although I guess it's fair to say, since you gave me the prompt, Lane, that my lab group will begin weekly water quality reports starting next week on News 12 on Thursday nights during the weather forecast. Those reports are water quality and more related to ecosystem conditions and ecosystem health. But we will be doing that preview next week, just before July 4th, and taking that through into Labor Day.
Excellent. All right, and here's a question from Mark. Does this work provide further insight into the ambient conditions that are conducive to blooms from year to year? Brown tide is physiologically adaptive. Why does that result in blooms in some years and not others?
Right, so very great question, and I think the answer is it's complicated, and I think the transcriptome partly shows that, right? Because I think again, we're looking, the organism itself is comprised of more than 11,000 genes that are clearly very, very active and turning on and off to deal with the dynamic ecosystem conditions. And on top of that, there's the external effect, or external biological effects like the grazers and the zooplankton. So, I think our work begins to give some insight with regards to all of the different factors that are impinging on the cell's physiology. And, I can't say we've got a robust answer to everything now, but clearly we're able to now identify and pick off the major organizing effects and the role of nitrogen, the role of phosphorus in conjunction with things that lead to cell mortality like the zooplankton grazing.
Okay, and from Teresa, we have a question. Have you looked at gene expression in response to changes in salinity and temperature?
No, we have not. We do know that brown tides often or typically end because of high temperatures. And, there is the opportunity with this project. In fact, we haven't actually gotten to do it. But during the decline phase of the bloom, we know that the temperatures got above the physiologically optimal levels for Oreococcus. So there is actually the opportunity to go in and look at genes related to temperature stress that may have been turned on as the bay got too warm.
All right. Just to close out, we have from Suffolk County, Nancy Pearson, who could address the issue of the question on beach quality. Nancy, I've unmuted you, so if you want to mention the beach monitoring program.
Hi, thank you. Can you hear me?
Yes.
No, I just wanted to address if they were interested in any water quality data for the beach program. We're currently monitoring, and it can be found at the link I sent there. Or you can actually go to suffolkcountyhealth.com, and in the search bar, you put beach monitoring program, and there's an interactive map that shows you the water quality data. But if you wanted a bigger subset of data, then you can just email me at my email there at nancy.pearson@countyny.gov. Yeah.
And I posted the web link to everyone in the chat, and I thank you for that. Thank you for that, Nancy. So I think we're gonna wrap up the presentation. I would like to remind you all to fill out our survey. I'm posting it again in the chat. It will help us with planning for the future of these seminars. Chris, it was an excellent presentation, and we appreciate you joining us for that. And we'd like to thank everyone who came and on Facebook and on this Zoom call. It was really glad to have you, and we hope we will see you at the next ones. Antoinette, do you want to add anything? Let me unmute you.
Thank you. I'd just like to echo your point, Lane. Thank you very much, Drs. Gobler and Dortch, for your presentations and the information that was presented. We are looking forward to, I'll say stay tuned to your continued research, Chris. And hopefully the next time we have a harmful algal bloom, we'll be able to meet in person. But until then, I would just remind everyone to please be safe. Remember to wash your hands or sanitize your hands, wear your face covering, so practice social distancing, because there is still this virus that's active, and we want you all to just try to practice safety as much as possible. And thank you again so much for taking the time to participate in our first seminar. And with that, have a great summer. Thanks, Lane.
All right, we hope to see most of you again in July. Thank you, Chris. Thank you, everybody. Have a good day.
Thanks, Lane. Thank you, everyone, for attending. Appreciated.